Bismillahirrahmanirrahim. 9700 June 24 paper 4-3 video 2 for question 5, 6 and 7. Now starting with question number 5, Bismillahirrahmanirrahim. BRCA2 is a tumor suppressor gene. Its gene product BRCA2 is involved in DNA repair. If the DNA cannot be repaired, BRCA2 has a role in causing the cell to die. BRCA2 is found in cells of breast tissue. When a mutation occurs in BRCA2, damaged DNA may not be repaired, and this increases the risk of breast cancer. When a double-stranded piece of DNA breaks, BRCA2 binds to the damaged DNA directly and interacts with the enzyme RAD51 to repair the damage. Repairing DNA prevents other mutations and gene rearrangements from occurring which could otherwise lead to breast cancer. Double-stranded DNA breaks occur naturally during meiosis. Double-stranded DNA breaks. These are sister chromatids. If crossing over has to take place between this one and this one, non-sister chromatids, what is that called? Crossing over. So it state the event that is initiated as a result of double-stranded breaks during meiosis, crossing over. Or you could have said chiasma formation, which always occurs in prophase 1. So just writing crossing over got you a mark for that. Then it says scientists have identified hundreds of mutations in BRCA2, but not all these mutations will increase the risk of cancer. One specific mutation in BRCA2 is known as 999DE15 is found in 0.6% of the general global population. Iceland is an island country in the North Atlantic Ocean. The ancestors of most of the current population are people who arrived to settle in Iceland in AD 874. In the Icelandic population, mutation 999DE15 is the cause of 7 to 8% of breast cancer cases in women and 40% of breast cancer cases in men. This is much higher than the percentage of breast cancer cases in the general global population. Suggest and explain how mutation 999DE15 accounts for a very high percentage of breast cancer cases in Iceland compared with the general global population. 3 marks. Mutation occurred a long time ago, or you could have said someone in the original population had the mutation. Mutation is a common ancestor. Then founder effect, then population is small. Much of the population is closely related, or you could have said a small gene pool. It's an isolated population, and this caused an increase in frequency of the mutation or the BRCA2 allele. So you have to know is that, if it is very high in this population, what is the reason for it? It's a very small gene pool, which is a low genetic diversity. So people who came to this island must be maybe say 100 people came, and now it's the same 100 people and their children and their children and their children. So it is just the same. So that's the founder effect. And the original population is small. Much of the population is closely related. It's an isolated population. We're not having many people coming in or people going out. If a lot of people were immigrating and emigrating, one is with an I and one is with an E, then things would have changed. So, suggest and explain. So those are the points that you would give for this part of the question. So mutation occurred a long time ago, founder effect, original population is small, or even if you said the current population is small, that would be fine. Small gene pool, or you can say low genetic diversity. It's an isolated population, increase in frequency of mutation. So reduce worry if negative, early treatment, breast removed, informed decision about having children, you could undergo genetic counseling, could lead to lower death rates from breast cancer. Then it says suggest one advantage to a country of genetic screening programs for breast cancer that screens for specific mutations in BRCA2 in the population. Now, if they only screen for mutations that occur with high frequency in a population, so it will be cost effective, early diagnosis or early treatment identifies mutations known to increase the risk of developing breast cancer. And of course, the same point as we said here could lead to lower death rate from breast cancer. So cost effective, early diagnosis, these were the points that we were expecting from you all. So cost effective and early diagnosis or early treatment and all the other points which I said as well. Then coming to part 5C, lipocalin-2 LCN2 is a cancer promoting gene, oncogene. When LCN2 is expressed, it can result in breast cancer. Research is being carried out to see if gene editing of LCN2 could be used to treat breast cancer. Gene editing was used to treat human cancer cells that had been implanted into mice to form a tumor. The treatment stopped LCN2 from being expressed in the cancer cells and resulted in a significant reduction in the growth of the tumor. There was no negative effect in normal tissues. Suggest how DNA editing stopped LCN2 from being expressed in cancer cells. You see, basically, you've got to understand if something is not working, what has happened? It is actually you have inserted or deleted or something, replaced something in the DNA. So the transcription of LCN2 is prevented. That protein which was promoting, which was resulting in the cancer, is not being produced anymore. Or you can say replace the mutated section of DNA with the correct sequence. Then it comes to the part 2 of the question. It says suggest why the expression of only LCN2 was affected. Only specific sites on the DNA is targeted. And that target site is only on LCN2. It's not anywhere else on the normal DNA, which is required for the normal cells of the organism. So specific sites are DNA targeted. Here in this part of the question, we're going to say only specific site or sequence of DNA are targeted, and those target sites only found in LCN2. So replace DNA in the LCN2 regulatory sequence. There's a regulatory sequence, there's a Y here. And so transcription of LCN2 prevented and replaced with correct sequence. Suggest why the expression of only LCN2 was affected. Only specific sequences on the DNA, which is only found in LCN2, was targeted. And coming to question number 6, in plants, stomata open and close in response to changes in environmental conditions. Explain why stomata need to open and close according to environmental conditions. Need to open to obtain carbon dioxide for photosynthesis. Why does the stomata need to open? We need carbon dioxide diffusing in from the air into the air spaces. So need to open to obtain carbon dioxide for photosynthesis, or you can say for the Calvin cycle. And need to close to prevent water loss by transpiration. Can close at night to save water as no photosynthesis is taking place. And must close to prevent water loss when there's high light intensity or high temperature, or there is high wind speed or water stress or drought conditions. So you could have said any of those things, 3 marks. Why do they need to open? Yes, because carbon dioxide has to diffuse in. And close to prevent water loss by transpiration, close at night when dark to save water as no photosynthesis taking place, and must close. Sometimes they also close in other situations like high light intensity will sometimes is not good for the plant. High temperature is not good. High wind speed, water stress, drought conditions. So they respond to that, abscisic acid is produced and the stomata close. So basically open to get carbon dioxide for photosynthesis, or you can say for the Calvin cycle, close to prevent water loss by transpiration, close at night as it is dark, so there's no photosynthesis, close during drought or water stress to prevent water loss, or high light intensity or high temperature or high wind speed, any of those points. Then of course we come to a very direct question. Describe the mechanism that's occurring in guard cells that leads to the opening of a stomata. And you can see this for 6 marks. This is for the 30 marks of the paper, 30 to 40 marks, which is direct questions. So the mechanism is hydrogen ions pumped out of the guard cell, decrease in hydrogen, and inside the guard cell, proton gradient setup, potassium channels open, potassium ions enter the guard cell, water potential decreases, water enters by osmosis, cells expand, outer wall is thinner so to allow more stretching, or you can say inner wall is thicker to allow less stretching, and then light activates ATP production. So proton pump is sort of a sentry for chloride ions. So you could have come so because the mechanism occurring that leads to the opening of a stomata. So basically we're talking about the opening. So it was 6 marks, but we have like 8 mark scheme points. So H positive pumped out by active transport, decrease in H positive inside guard cells, potassium channels open, potassium enters the guard cells, water potential decreases, water enters by osmosis, cells expand, outer wall to outer wall thinner to allow more stretching. Yeah, I missed that out. Outer wall thinner to allow more stretching. And the last point is light activates ATP production. So you could have said outer wall thinner to allow more stretching, or you could have said inner wall thicker to allow less stretching. You see, the guard cells have this inner wall and this outer wall. So the inner wall is thicker, this is thicker to allow less stretching, and the outer wall is thinner. So this expands more, and then that is how the guard cells open. You see, there's this and then there's this. So it's going to, so this is where the stromatal opening is inside here. Number 7. Experiments were carried out to determine the effect of light intensity on the rate of photosynthesis of a species of the unicellular Protoctus chlorella. A cell suspension of chlorella was used. Carbon dioxide uptake was used as a measure of the rate of photosynthesis. The suspension of chlorella was eliminated at light intensity 3 lux for 20 seconds. The carbon dioxide uptake by chlorella measured at the end of 20 seconds, period of elimination. The experiment was repeated at 6 lux, 9 lux, and 12 lux, and in a dark room. The suspension was maintained at a temperature of 20 degrees Celsius. Table 7.1 shows the results of the experiment. So we have 0, 0, 0, then we have 3, 20, 1.0, then 6, 4, 44, and 9, 72, 3.6, and 12. Use the table 7.1 to calculate the rate of photosynthesis at a light intensity of 6. So this is the thing that they want you to calculate. And then it says use table 7.1 to calculate the rate of photosynthesis at 6 lux and complete table 7.1. So you've got to write your answer here. So basically you had to calculate the rate, means this was in 20 seconds. So you can see here it was 20 seconds and here it is per second. So you just have to divide it by 20 and the answer was 2.2. Of course, you could have also guessed it from the other ones. From 20 it was 1 and 72 it was 3.6 and 80 it was 4.0. So you could have guessed it from that as well. So you wrote it in the space and you got a mark for that. Then we go on to the next part of the question. Then it says plot a graph of the data in table 7.1 on the grid in figure 7.1 to show the effect of light intensity on the rate of photosynthesis. Draw a curve and extend your curve to show what would happen to the rate of photosynthesis if the experiment was carried out at 18 lux. So you would draw a curve and then you can see you can have tried to plot it. I might not have done it very accurately, I'm sorry. But then you connect the lines and then you connect this line and then you connect, now you see it's going to increase, so it's going to become something like this. So it said extend it to 18 lux. So I've extended it to 18 lux. I've given it a small curve, continues up but a smaller gradient. And suggest an explanation for the shape of your curve from 12x lux to 18 lux. So between 12 and 18, you're going to give me an explanation for that. Now, explanation is because light intensity is no longer limiting. Temperature or carbon dioxide concentration is now limiting. So that is why we sort of see there's not much of an increase left, because as you can see, the gradient is decreasing of the rate of photosynthesis. Because light intensity no longer limiting, now temperature or carbon dioxide concentration is limiting. So we have the three things which limit photosynthesis: TLC, temperature, light intensity, and CO2 concentration. Now coming to the B part of the question. In photophosphorylation, photoactivation of chlorophyll results in the synthesis of ATP. Describe how the photoactivation of chlorophyll results in the synthesis of ATP in photophosphorylation. So electrons excited, electrons travel along the ETC and energy released. Then the energy is released is to pump hydrogen ions into the thylakoid space. Increased concentration of hydrogen protons in the thylakoid space creates a proton gradient. Then the protons diffuse through the ATP synthase into the stroma, and this is chemiosmosis. So electrons excited, electrons travel along ETC and energy released to pump hydrogen ions into thylakoid space. Increase protons in thylakoid space, proton diffuse through ATP synthase to stroma, and this is called chemiosmosis. This is exactly what we are trying to say. You see, the electron transport chain is the one photosystem 2 and photosystem 1. You know, when the electrons are being excited and they're going to be moving from here to here, from photosystem 2 to photosystem 1, then electrons are excited. Electrons travel along ETC and energy is released. Then this energy is used to pump hydrogen ions into the thylakoid space. You can see this in red here. The thylakoids being pumped into the thylakoid space. You see, this is the inside, the whole thylakoid membrane. You can see this is the thylakoid membrane, and outside is the stroma. So the hydrogen ions are all pumped into the thylakoid space, and then the hydrogen ions have nowhere to go out, but then the only way they can go out is through the ATP synthase. And that is why now they're going into the stroma. Because in the stroma, then what is going to take place is the Calvin cycle. So you've got to understand this. I'm sorry, this is part of the syllabus, and the new syllabus we've got these things. We didn't have this in the older syllabus. So hydrogen ions increase concentration of protons inside here. Now there's more in the thylakoid space. Increased concentration of hydrogen ions or protons, and then the hydrogen or the protons diffuse through the ATP synthase from the thylakoid space to the stroma. This is the stroma here. This outside is the stroma. Thylakoid space, the outside thing around it is the stroma, and this is called chemiosmosis. So I've just revised it with you. I hope everybody's clear on this. And you see the water splitting reaction, you can see that here, the photolysis of water taking place here. Thank you, and we will continue the rest of the questions in the next video.